Bismillah Rahman Rahim. This is the 9700A2 Bio November 22 question paper 5-2, and this is the question 2 which has been explained in this video. Now coming to question number 2, that is now the first question was for 15 marks, so this is for another 15 marks. Now this question is a little different than previous papers as I see. A main feature of Huntington's disease HD is the loss of neurons from a region of the brain known as the striatum. A study of brain tissue taken from people with HD showed higher than normal increased urea concentrations in the anterior striatum and cerebellar regions of the brain. Basically, more urea in the brain. Which parts of the brain? Well, that is, of course, the names have been given to you. But please remember the structure of the brain is not in the syllabus. Urea is a toxic waste product of amino acid and protein metabolism. It is produced from ammonia, which is also toxic in an enzyme-controlled cycle known as the urea cycle. Scientists carried out a study using transgenic genetically modified sheep known as OVT73 to investigate the increased urea concentrations. OVT73 have the mutant allele of the Huntington's gene, HTT. At the time of the study, OVT73 were 5 years old and had no detectable loss of striatal neurons or disease symptoms. Samples of tissue were taken from the same region of the anterior striatum in 6 OVT73, and only 6 sheep were 3 males and 3 females, a control group of non-transgenic sheep. Describe 2 features of an appropriate control group for this investigation. Well, they should have been the same gender, they should have been 3 males and 3 females, and they should have been the same age, or you could have said 5 years old, and they should have been the same breed or the same type of sheep, then the same size or the same mass, and the fact they did not have the mutant allele or the Huntington's disease. So whenever we're talking of a control group, it has to be a very fair thing. So if we're going to do it on, we must have the same number, we must have them the same age, they must be the same species or the same type, and they must be the same size, because that is also going to affect the results. Then coming to the B part of the question, a whole genome RNA sequencing experiment was carried out on the tissue samples using a commercially available diagnostic testing kit. This experiment produced gene expression profiles for comparison. From the hundreds of genes that showed differential gene expression, 24 genes were described as genes of interest because they were involved in urea metabolism or in urea transport out of cells. None of these were genes coding for enzymes in the urea cycle. The 24 genes of interest were identified from RNA sequences using mRNA transcripts. Explain why it was more appropriate to use RNA sequencing rather than DNA sequencing for the identification of these genes of interest. You see, the DNA contains many genes. So DNA sequences will only identify the presence of genes. But what does the RNA sequencing tell you? The RNA sequences identifies which genes are being expressed, or you can say which genes are being switched on. Now, for instance, this is the nucleus. Now, inside you've got a lot of DNA. But if I say, okay, there is this mRNA, there is this mRNA, there is this mRNA, so you know 3 proteins are being made. But the DNA just tells us is okay, there are genes present, but it doesn't tell us which gene is being expressed. So if we know that there are 3 types of mRNA, then we know that 3 types of protein are being made. So it says why it was more appropriate to use RNA sequencing rather than DNA sequencing. The reason is the quantity of RNA indicates the level of gene expression. So the more the copies of the mRNA, then more we know that that certain protein is being made. So RNA sequencing identifies which genes have been expressed, or you can say which have been switched on, while the DNA only identifies the presence of genes. So DNA identifies presence of genes, RNA identifies expressed genes, quantity of RNA indicates level of gene expression. So part C of the question, a second sample of tissue was taken from the same region of the anterior striatum of each of the same individuals. A different commercial diagnostic testing kit was used to obtain a comparison of gene expression for the 24 genes of interest. Analysis of the results identified 10 genes with differential gene expression at a statistically significant level of probability less than 0.05. Table 2.1 shows the results for these 10 genes and how much greater the value of OVT73 is calculated as a ratio. Mean count is a standardized value based on the quantity of mRNA transcripts obtained from the diagnostic testing. SE is standard error. So we have this table, table 2.1, which is a very complicated table. And the name of the genes are given here. And then we have OVT73 mean count and the control mean count and the standard error and the ratio. Ratio is the OVT73 count divided by the control count. So this is 2.20, 2.18, 1.82, 1.53, 1.49, 1.47. So you look at this very carefully. You look at all the standard errors and then you look at this one as well, the mean count. Now it says state which statistical test would have been used to establish the statistical significance between the control and the OVT73 data sets. Explain why the test was chosen. Now, whenever we are discussing two sets of data, that is always a t-test. So that is something we all know, that it is a t-test. And why are we doing the t-test is because it is comparing the means. So whenever we are doing the t-test, it is comparing the means, or you can say continuous data, or comparing two samples with fewer than 30 values. Now it says state the null hypothesis in this investigation. Now this is for 1 mark, and this is nobody should really lose this mark because it's something which we all know. Null hypothesis means when you negate the hypothesis. So there is no difference in the mean counts between the OVT73 and the control, because that is what we were comparing in the previous table. If you look at that, you see what were we doing? We were comparing the OVT73 mean count and the control mean count. So the null hypothesis is always you negate it. There is no difference between the mean counts in OVT73 and the control. Null hypothesis find no difference in the mean count between OVT73 and control. With reference to table 2.1, explain the advantage of calculating the ratios. Now, you see here the ratios were here. Is OVT73 count divided by the control count. Now, if you look at it, just look at it. How did we do this? So you give yourself examples. 10 over 2 is equal to 5, 10 over 10 is equal to... What does the ratio tell you? The ratios tell you it's very easy to make a comparison. If I say, okay, 10% of the students in school A, I teach in school A, and the 10% of the students got an A. And in the other school, 5% got an A. So it's a very good way to compare. We don't know the number of students, but if I give you a percentage or something like a ratio, it's a very good way to compare the data. Now, if I say two students got an A, well, you have no clue. But if I say 10% of my students got an A, then you get to know that if she had 100 students, 10 only got an A. So to make a comparison, the ratios are a very good way to make a comparison and also to identify if the OVT73 mean count was higher or lower than the control, because you're comparing it with the control. So this is a very good way. The ratios or the percentages is a way to compare. So to make a comparison and to identify if OVT73 count was higher than the control. Or you could have used a data from the table 2.1 to support this statement of yours. Then coming to the D part of the question, the results of the two RNA sequencing experiments indicate that there is a difference between OVT73 and the control group in the expression of each of the 10 genes shown in table 2.1. State one feature of the study that contributes to the validity is valid of these results and one feature that would improve the validity of these results. So feature that contributes and improve, feature that contributes, two different testing kits used. Then commercial kits used, so they are accurate. Then statistical test was carried out. That all improves the validity. And for improvement, of course, this is something which will come in every paper 5, that if you've used a very small number of experimental data, like here only a very small number of sheep were used, only 6 sheep were used. So more sheep will, of course, make it more valid. So more sheep, or you can say more tissue samples, that is something which can always write in improving the validity of the results. So contributing to validity, commercial tests used, they were more accurate. Then the statistical tests were carried out, and two different testing kits were used. But improving the validity, we should use more of the sheep, we can use more of the tissue samples, or we can test the same sample again and again. So we should take many samples from the same tissue as well. You could have even said that. Then coming to the part E of the question, differential expression of the 10 genes shown in table 2.1 was compared for tissue samples taken from a different region of the anterior striatum. Only gene SLC14A1 showed differential gene expression at a significant level. This gene also showed statistically significant differential gene expression in the cerebellum and the motor cortex region of the brain. The scientists then measured the concentration of urea in tissue samples taken from these three regions of the brain. The results are shown in table 2.2. So there's a very complicated table 2.2 in which we have the mean urea concentration and the standard error in nanomoles dm cube urea milligram per milligram of protein, and then we have the ratio, and then we have the probability value 0.02, 0.22, 0.31. Complete table 2.2 by calculating the ratio for the motor cortex. Now this is 1 mark. Now there's something you need to write here. Now if you get this wrong, this is that very easy 1 mark which everybody should get. So very simple, you had to give me the, like it says here, 1.54, 1.75, so you must also give it to 2 decimal points. So it was 1.33. This divided by this got you this answer. Then with reference to table 2.2, explain how the standard error, where is the standard error? Let's look at the standard error. This is the standard error. This is the standard error. Here. So with reference to table, to explain how the standard error values for mean urea concentration are good indicators of the estimates of statistical significance. So with the reference to table 2.2, explain how the standard error values is the estimates of, you see, you have to understand for the anterior striatum, the standard error ranges do not overlap, suggesting that there is a significant difference. If they do not overlap the standard errors, then there is a significant difference. But for the cerebellum and the motor cortex, these ones, this and this here, they do overlap. So there is not a significant difference. So when they do overlap, there's not a significant difference. When they do not overlap, there is a significant difference. So for the anterior striatum, the standard errors 35.2 and 8.6, they do not overlap. So there is a significant difference. But for the cerebellum and the motor cortex, this is 17.5, 19.8, 10.3, and 12.5. They do overlap, so there is not a significant difference. So when they do overlap, not a significant difference. When they do not overlap, there is a significant difference. SLC14A1 codes for a transport protein that functions in the facilitated diffusion of urea out of cells. So, okay, this is a cell. This is a channel protein, and this allows urea to move out. Always do this diagram in your mind. So SLC14A1 codes for a transport protein that functions in the facilitated diffusion of urea. So urea is more outside, less here, facilitated diffusion. Explain the relationship between the results shown in table 2.2 and the increased gene expression shown by SLC. What was very simple. More urea in the cell. So more transport proteins, more removal of the urea. More urea in the cell. So if there's more urea in the cell, there'll be more transport proteins, and more of the urea will be moved out of the cell because it's toxic, it's bad for the cell. In fact, when the person suffers from kidney failure, he goes into uremic coma. It's the brain cells which are affected. So more urea in the cell, so more transport proteins and more removal of the urea. So 1 mark, more urea in the cell, more transport proteins, more removal of the urea. So basically you just write the question again. You just write what was information was given in the question. You just had to register that and write that in a way you have to write that again. So that is what is common sense. Of course, common sense is the rarest sense. Coming to the last part of the question, the results of the study show a relationship between the increased urea concentrations and Huntington's disease, but also highlight the need for further investigation. Some possible causes of increased urea concentration in the cells are a changed expression in the gene coding for the enzymes of the urea cycle, an increase in protein breakdown related to cell death, an increase in breakdown to provide more energy to a cell that has an increased rate of metabolism. Explain why the results of this study indicate that possible causes 1 and 2 are less likely to be involved in the early stages of HD than possible cause 3. And there's 2 marks. Now that finishes your question. Now, how are you going to go about this question? As I always say, read the question again once you read the whole question, the information given to you. Now, here it was very categoric. None of these genes coding for enzymes in the urea cycle. But you read it earlier. But then you're not going to remember this when you're reading the next part of the question, which was the last part of the question. So that is the first mark scheme point. None of the genes coded for enzymes in the urea cycle. So actually we are writing some information which was given to us in the question. And we are writing that again in that answer, which was the last part of the question, which was the answer here. Why the results? Because they said here coding for the enzymes of the urea cycle. Now that was given in the question which I've just shown you. So none of the genes coded for enzymes in the urea cycle. No loss of neurons in OVT73. More urea produced, so more transport proteins made. That means the gene expression, so cells require more energy. So that's how we word it. None of the genes coded for enzymes in the urea cycle, no loss of neurons in OVT73. More urea produced, so you need more transport proteins. You know, for protein synthesis, you need more energy. So cells require more energy. And that was the point that they gave here, provide more energy here. This is the point that they were giving here. So basically, paper 5 is reading the question very carefully, going back, rereading the question, looking at the questions which have come, and taking your time and spending the 1 hour 15 minutes in very, very careful, fruitful contemplation and thinking about the answers and then writing your answers. So this was another total 15 marks, and this was the most difficult paper as I just discussed with you in the beginning, and which had the lowest marks for the A grade. So most of the students got it wrong. So you better try your best at the paper which is going to come tomorrow. Wish you all the best, and thank you all for subscribing, and thank you for watching these videos, and thank you for leaving all the very good comments. Thank you very much.